Good day, everyone. So we're reviewing Wetzel's Limnology textbook. This is edition number 4, and we are on chapter 14, the nitrogen cycle. And more specifically, actually, today we'll be talking about nitrogen cycling, and this is where we left it at yesterday, right here. Compared to lakes and reservoirs, hydrological regimes can have strong impacts on nutrient dynamics, especially for streams and rivers in arid, semi-arid, and Mediterranean areas. Intermittent rivers were conservatively estimated to account for 30% of the total length and discharge of the global river network and are common around the world, especially in Australia. Intensive water abstraction and regulation, as well as climate change, can also shift flow regimes and results in disrupted flows in streams and rivers. Nutrient sources, in-stream nutrient processes, and hydrological interaction between the surface stream, hyporheic, parafluvial, and riparian zones can differ significantly between dry, wet, and transitional phases in intermittent river systems. Cessation of surface water flow causes rivers to retract, and disconnected water holes, such as kettle holes, might emerge. Persistent water holes can have high primary productivity during dry periods and become hotspots for nutrient processing when flow reconnects. Nitrate concentration in water holes can decrease due to denitrification, which is enhanced by hypoxic conditions. In contrast, increased ammonium concentration mainly resulted from the rapid mineralization of accumulated organic detritus, or enhanced DNRA, and inhibited nitrification processes in hypoxic conditions. The impact of desiccation on nitrogen processes in the dry sediment has been shown to vary substantially between studies. For example, nitrification and denitrification have been shown to increase, decrease, or remain unchanged in dry sediments of intermittent rivers, depending on the study. This is probably due to differences in desiccation, duration, and subsurface flows. Large amounts of nutrients can be transported from catchments into rivers during these first flush events. The in-stream nitrogen processes, such as denitrification and nitrification, can be activated rapidly upon rewetting, together with the recovery of nitrogen retention in streams and rivers after the first flow event. So before we move on to the next chapter, let's look at this figure here. Figure 14-13, the cycle of nitrogen in river systems, sources, transformation, and flux. So we have the sediment here, and the sediment is subject to transport and transformation with these ionic species, as well as nitrous oxide and dinitrogen. We have a release mechanism from the overlying water into nitrogen, dinitrogen, and nitrous oxide. Then we have denitrification, nitrification, anammox, and then we have the nitrogen cycle. And then water conservancy projects has an influence upon the water sediment conditions. Human activities, so we have regulatory activities that will, that basically drive some of the water conservancy project. And then domestic waste, such as wastewater treatment plants, outflow, and that may be secondarily treated but may not have the nitrogen removed from it. And then human activities also has an influence upon the nitrogen cycle in the terrestrial system with nitrogen deposition, livestock manure, nitrogen fertilizer, and that has an input upon to the rivers system. Then we have climate change, with a change in precipitation patterns, change in temperature, change in light regimes, change in wind speed, which will have an influence upon the overlying water and the nitrogen cycle with these three processes that we already went through. And then the hydrological conditions, runoff such as soil leaching, will also have an influence upon the nitrogen inputs to rivers. Effect of human activities, so eutrophication. Enrichments of freshwaters with nutrients commonly occurs as a result of catchment losses from agricultural fertilization loading from sewage and industrial waste, and enrichment via atmospheric pollutants, especially nitrate and ammonium. Excessive loading of these nutrients permits increased plant growth, including algal growth, until other nutrients or light availability ultimately limit further plant biomass accumulation. The pathways of anthropogenic nitrogen inputs to freshwater can be seen in figure 14-13, which is what we just reviewed. At the catchment scale, nitrogen fertilizer application for agriculture is a major contributor to eutrophication. It is estimated to double the amount of bioavailable nitrogen to the terrestrial biosphere annually. The tropics are a major contributor, producing 56 plus or minus 6% of the global land nitrogen pollution, despite covering only 34% of the global land area. Over the last few decades, ammonium nitrate synthesis has been replaced by the chemical synthesis of urea, and it is now the most widely used nitrogenous fertilizer. Studies have shown that urea concentration can be more than twofold greater in lakes receiving nitrogen from cities than in agriculturally affected basins. Furthermore, the nitrogen-phosphorus ratio of global fertilizer use has increased over time. This is likely to have altered the ratios in inflows to freshwater system, with resultant effects on biogeochemical processes. Increased nitrogen loads to waterways can stimulate excessive algal productivity, negatively affecting the health of aquatic ecosystems. Increased nitrogen loading also reduces microalgal and aquatic plant biodiversity, changes the population structure, and is associated with the eventual loss of macrophyte communities. The subsequent decomposition of lost macrophytes could be an important internal nutrient source for the water column. Historically, phosphorus was considered the key nutrient controlling algal growth in freshwater systems. This information has been used to determine the effect of phosphorus loading on productivity in lakes. However, in recent years, the phosphorus limitation paradigm has been challenged, with arguments made that controlling only phosphorus may not be sufficient for reducing productivity in some lakes or reservoirs. This may be because nitrogen fixation as an external source of nitrogen cannot always compensate for nitrogen loss. Nitrogen may also escape via denitrification, leading to perpetual nitrogen deficits. Thus, dual nitrogen and phosphorus reductions may be needed to reduce harmful algal blooms in many lakes and reservoirs. Global analysis have shown that despite differences in marine, terrestrial, and freshwater habitats, significant synergistic effects of combined nitrogen and phosphorus enrichment are seen across many ecosystems. An important step in minimizing eutrophication is to reduce external nitrogen loads to waterways. However, nitrogen release and cycling across the sediment-water interface within freshwater system is also an important mechanism leading to eutrophication. In lakes or reservoirs in which external loading has been reduced, internal nutrient loading may prevent the reduction of eutrophication. Increased atmospheric nitrogen deposition in lakes as a result of anthropogenic activities has also contributed to eutrophication. A global analysis has shown that increased atmospheric nitrogen deposition may have shifted some lakes from nitrogen to phosphorus limitation. This impacts nitrogen cycling and higher trophic levels. The impact of long-term increases in anthropogenic nitrogen has been shown in sediment and fish in Lake Ontario. The nitrogen stable isotope was used to reflect transformation processes with an increase in the ratio of the heavier nitrogen-15 isotope around the 1830s, at a time when industrial-scale forestry increased. Since that time, the ratio of nitrogen-15 isotope has continued to increase. There are a range of likely sources of increased nitrogen runoff, including industrial-scale forestry and broad-scale clearing for agriculture. Warmer temperature, so let's make sure we don't, here we go, climate change, Climate change can also impact nitrogen budgets and transformation in freshwater systems. Warmer temperatures increase enzyme activity affecting rates of microbial nitrogen transformation until a temperature optimum is reached. Climate change is also expected to intensify the hydrologic cycle, including changes such as more frequent and intense heavy rainfall events, potential deepening and lengthening of dry periods, altered snow accumulation and melt, and changes in evapotranspiration. This will affect the rates of microbial nitrogen transformation such as decomposition, mineralization, nitrification, denitrification, and biological nitrogen fixation. Climate-driven changes to the hydrologic cycle will also alter the quantity of nitrogen transported through a system via waterborne transport and soil water content mediated nitrogen cycling. Greater precipitation generally increases water flow, which may increase leaching, export of nitrogen through terrestrial landscapes, increase terrestrial nitrogen inputs to streams and rivers, and increase nitrogen transport rates through streams and rivers through adaptation by microbes and plants may increase their ability to retain nitrogen as flushing increases, thereby potentially limiting some of the overall impact of increased precipitation and flow. Beyond simply the total volume of precipitation, precipitation intensity influences the rate of nitrogen flow through ecosystems and affects nitrogen spiraling rates. Increased precipitation intensity of storm system, as is expected to continue with global warming, would likely increase the frequency of high nitrogen loading events to aquatic systems. Due to the limited capacity for in-stream removal of nitrogen during high flow pulse events, most nitrogen is transported downstream. A study showed that North European temperate lakes, for example, have projected increases in winter rainfall and higher agricultural cropping practices, which will increase runoff and nitrogen transport annually. This, combined with higher temperatures, may increase the risk of cyanobacterial blooms, including harmful blooms in lakes and reservoirs. In contrast, in warm, arid lakes, increased temperatures will increase evapotranspiration, concentrating nutrients in the shallow systems and exacerbating water quality issues. Such hydrologic cycle changes are also expected to affect the timing of nitrogen transport. Changes in the seasonality of precipitation, and in particular snowmelt, will tend to alter the timing of nitrogen flushing through the ecosystem. There have already been widespread instances of earlier snowmelt and increased winter thaws associated with warming over the past few decades. Timing changes can ultimately alter the magnitude of nitrogen export to downstream water bodies, particularly if the timing of flushing changes relative to the timing of biologically mediated uptake in either terrestrial or aquatic ecosystems. So that will complete our reading of nitrogen cycling. Why don't we go quickly over this summary, and we should be done for the day. So this is the summary of the chapter 14. Nitrogen, along with carbon, hydrogen, and phosphorus, are major constituents of the cellular protoplasm of organisms. Nitrogen is a major nutrient that affects the productivity of freshwaters. The nitrogen cycle is a complex biochemical process in which nitrogen in various forms is altered. This includes nitrogen fixation, assimilation, remineralization, anamox, and denitrification, to name a few. For all practical purposes, the nitrogen cycle of lakes and rivers is microbial in nature. Bacterial oxidation and reduction of nitrogen compounds are coupled with photosynthetic assimilation and utilization by algae, cyanobacteria, and larger aquatic plants. The direct role of animals in the nitrogen cycle is typically very small. Dominant forms of nitrogen in freshwaters include dissolved molecular nitrogen, ammonium, nitrite, nitrate, and a suite of organic compounds, for example, amino acid, amine, nucleotides, protein, etc., in particulate nitrogen. The nitrogen budget consists of a balance between nitrogen inputs to and nitrogen losses from an aquatic ecosystem. Sources of nitrogen include atmospheric, nitrogen fixation, inputs of nitrogen from surface and groundwater drainage. Losses of nitrogen occur by outflow from the basin, denitrification and anamox, and sedimentation of inorganic and organic nitrogen containing compounds to the sediment. Microbial fixation of molecular nitrogen in soils by bacteria is a major source of nitrogen. In lakes and streams, nitrogen fixation by heterotrophic bacteria and certain cyanobacteria is quantitatively less significant, except under certain conditions of severe depletion of combined inorganic nitrogen compounds. Nitrogen fixation by cyanobacteria is usually much greater than fixation by heterotrophic bacteria. In cyanobacteria, nitrogen fixation is light dependent and usually coincides with the spatial and temporal distribution of these microbes. Ammonium assimilation requires less energy expenditure than nitrogen fixation by cyanobacteria, and hence nitrogen fixation increases when ammonium and nitrate concentration decrease in the trophogenic zone. Ammonium is generated by heterotrophic bacteria as the primary nitrogenous end product of the decomposition of protein and other nitrogenous organic compounds. Ammonium can also be generated in anaerobic conditions via dissimilatory nitrate reduction to ammonium, or DNRA. Ammonium is readily assimilated by plants in the trophogenic zone. Ammonium concentrations are usually close to detection limits in aerobic waters because of utilization by plants in the photic zone. Additionally, bacterial nitrification occurs, resulting in ammonium being oxidized through several intermediate compounds to nitrite and nitrate. When the hypolimnion of a eutrophic lake becomes anaerobic, bacterial nitrification of ammonium ceases. The oxidized macrozone at the sediment water interface is also lost, which reduces the adsorptive capacity of the sediment for ammonium. A marked increase in the release of ammonium from the sediment then occurs. As a result, the ammonium concentration of the hypolimnion increase. Bacterial nitrification normally proceeds in two stages: the oxidation of ammonium to nitrite, largely by ammonium oxidizing archaea, and ammonium oxidizing bacteria. Oxidation of nitrite to nitrate, in which nitrate oxidizing bacteria are involved. However, the recent discovery of complete ammonium oxidizers, Comamox, showed that the oxidation of ammonium to nitrite can also be done by a single macroorganism. Nitrite is readily oxidized and rarely accumulates except in the metalimnium, upper hypolimnion, or interstitial water of sediments of eutrophic lakes. Concentrations are usually close to detection limits. Nitrate is assimilated and aminated into organic nitrogenous compound within organisms. This organic nitrogen is bound and cycled in photosynthetic and microbial organisms during the normal metabolism of these organisms, and at death, much of their nitrogen is liberated as ammonium. Additionally, organisms release a variety of organic nitrogenous compounds that are resistant to proteolytic deamination and ammonification by heterotrophic bacteria to varying degrees. Nitrate is the common form of inorganic nitrogen entering freshwaters from the drainage basin in surface waters, groundwater and precipitation. In certain oligotrophic waters in basaltic rock formation, nitrate loading from atmospheric sources, especially if contaminated by human-produced combustion emissions. Bacterial denitrification is the biochemical reduction of oxidized nitrogen anions, so nitrate to nitrite, concomitant with the oxidation of organic matter. So we have nitrate to nitrite to nitrous oxide to dinitrogen. Nitrous oxide is rapidly reduced to dinitrogen, as has rarely been found in lakes in appreciable quantities. Denitrification is accomplished by many genera of facultative anaerobic bacteria, which utilizes nitrate as an exogenous terminal hydrogen acceptor in the oxidation of organic substrates. Denitrification occurs in anaerobic environments such as the apolimnia of eutrophic lakes or in anoxic sediments where oxidable substrates are relatively abundant. The process of anamox has only been described relatively recently. This process converts 1-to-1 molecular ratio of ammonium and nitrite to dinitrogen in the absence of oxygen and without the need for a carbon supply. The process of dissimilatory nitrate reduction to ammonium, DNRA, also contributes to nitrate removal in aquatic ecosystems, but the relative importance of DNRA and denitrification on nitrate removal is quite variable between different aquatic systems. Dissolved organic nitrogen often constitutes over 50% of the total soluble nitrogen in fresh water. Much of the total nitrogen occurs in the sediments in the form that are relatively unavailable for biotic utilization. Of the readily available nitrogen, a majority occurs in soluble forms in the water and the interstitial water of superficial sediments. Turnover rates of ammonium are rapid in water but slower in the sediment. In contrast, nitrate turnover is slower in the water than in sediment, where under anoxic conditions in eutrophic lakes, nitrate may be denitrified. In running waters, nitrogen is used repeatedly as it passes downstream. The rate of utilization and release depends upon physical and biological retentiveness, largely by the microbiota attached to the stream bed. The average distance a nutrient atom travels downstreams during one cycle through the water, biotic, and substrata compartments is referred to as the spiraling length. Water flow in streams and rivers can continuously bring dissolved nutrients to primary producers, especially algae, attached algae, and other plants. However, nutrient limitation often occurs in streams and rivers in the absence of excessive nutrient inputs. The process of nitrogen cycling in the water of streams and rivers are similar to those of lakes and are influenced to a large extent by bacterial, fungal, and other microbial metabolism. Attached bacteria, fungi, and algae are the primary organisms controlling the seasonal, spatial, and temporal variation within the water. The processes of nitrification and denitrification often function simultaneously and reciprocally in running water sediments, where many macrozones of steep redox gradients occur in the aporeic zone of the stream bed. Increased loading of inorganic nitrogen to rivers and lakes results from agricultural activities, sewage, and anthropogenic atmospheric pollutions. Increased nitrogen loads to waterways can stimulate excessive algal productivity, reduce plant biodiversity, are associated with loss of macrophyte communities, and have effects on the quality and quantity of food available for aquatic fauna. Fertilizer use for crops is a major source of nitrogen to freshwaters globally, and is changing the ratio of nitrogen to phosphorus with impacts on the functioning of aquatic systems. Climate change can also impact nitrogen budgets and transformation in freshwater systems. Warmer temperatures increase enzyme activity, affecting rates of microbial nitrogen transformation. Climate change is also expected to intensify the hydrologic cycle with more frequent and intense heavy rainfall events, potential deepening and lengthening of dry periods, altered snow accumulation and melt, and changes in evapotranspiration. This will affect the rates of microbial nitrogen transformation. And that is it for chapter 14, nitrogen cycle. This is the reference area, and let's look at chapter 15 that we're going to be starting tomorrow, and that is going to be the phosphorus cycle, chapter 15. So I'll see you tomorrow.